Thanks. I'll invite the speakers to come back up, and we'll take some questions. Go ahead.
Kapil Bhati from NIH. I had to ask the first question. So great talks, but three of you all had different doses of cells or patch you are transplanting, all for AMD. So Sally had about 50 to 150,000 cells and had 200 to 1 million cells, and you have a patch. Can you each comment on the choice, why you picked the dose you picked, and the rationale behind that?
Thanks. Thanks, Kapil. So the area, the diseased area is relatively small. That on the order of a few thousand cells should, in theory, cover that and replace the cells that are lost. And so that is where we came with our dosage range. Also, ACT had used a similar dosage range previously. So we did use that information, but I do want to underline what Lorenz said earlier, which is that you begin to make decisions on best guesses, frankly, because these are things that you have to determine empirically in the patient. And so, you know, we do base that on past experience and, of course, the animal modeling that we've been doing.
So we also based our doses on A, the target area, and B, the animal models. So for example, in the brains for Battens, we did from 500 million to 1 billion cells because that is a neurodegenerative affecting the whole brain. The brain is quite large. You wanted to deliver as many cells as quickly as possible. In the case of the retina, it was a much smaller area, and the cells migrate, so we wanted it to cover a larger area, and so we ended up with a dose of 20 million. For spinal cord, because you know, if you cause any harm in the spinal cord, you could cause more damage to the paralyzed patients. And so in the first trial in thoracic, we did 20 million cells. In the cervical, we started from 15 to 30 to 40 to show that we weren't causing any damage in that case. And again, it was allometric scaling on the volume of the cord compared to the cord in the animal models.
So we based it on patient ability to read. So the 3 by 6 millimeter area within the macula allows a visual angle which allows a person to be able to read, and therefore the limiting factor was the number of cells then that actually grow on that patch.
So actually, just expanding on that, can you three comment on what's the predicted mechanism of action based on this dose that you chose for, you know, small number of cells, very large number of cells, then same number of cells but a patch? What is the mechanism of action you're predicting from, or hypothesis from your engraft?
So ours is true replacement of the RPE. So it would be fully functional RPE.
Cindy Novartis, interested in you guys talking about both the eye and the brain in terms of especially working with allogeneic, fully allogeneic or totally mismatched cells. I mean, how long do you really think you're going to get permanent tolerance, especially in a setting where there's ongoing inflammation if you don't have, you know, sort of perfect efficacy right away? I mean, as a transplanter, you know, hematopoietic stem cells eventually do lead to tolerance because you make new T cells and they become, but you know, in solid organs, you have to stay on immunosuppression for life. And I'm just interested as to why and when you decided to stop, or was, you know, you said it might be too early, but I think to me it would always be too early if you have any ongoing inflammation. So anyway, I'm just interested as to what models you use to try to work this out, because mice have been so bad and xenografts of human cells and monkeys aren't so great either. So Anyway.
So we asked the immunologist what we should do, and we never got a straight answer. Apart from, well, it's an organ, really, so therefore it should be triple and immunosuppression. So we said no, we weren't going to do that. So in fact, the patients which we operated on were of the wet kind, so the blood-retinal barrier was gone, completely gone. And these were really bad cases. All we use is the typical anti-inflammatories you would use for surgery. So pranicillone initially, and then a triamcinolone, which just gives you a 3-month period. But what we have done is we've actually put a capsule which secretes steroids into the back of the eye, which can last 2 years. So I think the problem is I don't think there is a good model. We tried very hard. We did look at bloods from AMD patients and how there was any reactivity to the RPE cells themselves, and we didn't see any. But I mean, the RPE again is a unique cell. It actually knocks down the T cell response.
So, you're right. There's no good model to look at graft versus host, or tolerance induction. And one of the points that I didn't get to is that, if you really do want to have tolerance to your graft, or you really want to achieve tolerance to your graft, you might want to be able to co-transplant hematopoietic stem cells and your organ through ES-derived cells or IPS-derived cells, but that's a long way off. Well, yes, and so, we looked at the data that had been published in the Parkinson's where they had the fetal tissue grafts, and we looked, and so it was a best guess, but as I mentioned, going from the thoracic trial at 9 months to the cervical trial at 6 months, as I mentioned, the thoracic, you can argue that it's the patient population, or you can argue that it's a difference between the thoracic cord and the cervical cord, but, shortening the length of immunosuppression might also have been a factor in this. And so we can't address that question unless we go back and repeat it with a similar patient population, going to back to the 9 months as we did in the thoracic. So
I was curious again because there are now so many basically efforts going forward with the RPE, and we heard this morning a little bit how at least in other field, like in Parkinson's, another area, we try to kind of have consortia or interaction and exchanges, and I was curious because you guys are kind of ahead, the companies are involved at that stage, and how much is that still in play, or is it really getting much more kind of locked down, or is there really ways that you guys really exchange data early on, warn each other? Is that something that's still kind of in play at that stage?
I think that would be wonderful, and we'd be very open to it, Lorenz. There's a lot of different entities involved, and ensuring that they will share information is key. We were talking about that in the broader context of providing the information about what happens in clinical trials, including the failures, including the negative results, so that we don't repeat those mistakes. Having better access to the clinical data, all of this would be enormously useful. But there has to be some incentive to create the consortium. I believe this would be, because ultimately it ensures that we use what are really limited resources more efficiently.
I would say there has been a good spread of information from those involved in these types of trials. Personally, I've been involved with California and the project which is happening there, and also other projects which are on the receptor side, which was a trial which has just started in Harvard. We did some of their preclinical talks for them. So I think it is there. There is an issue, obviously. There's Pfizer, who are involved with the London project. There's only so much information there that I can give on the process. But as you saw, we're more than open to show the process we've been through in terms of that clinical trial application. And so has Pfizer. Pfizer's been very, very aware that this is important information for the greater cellular community, not just for the RPE.
David Barron, PKD Foundation. I'm curious whether you think that the regulatory burden imposed was appropriate for the seriousness of the disease, and more particularly from a patient perspective, I think the benefit-risk ratio might be somewhat different than the regulatory agencies who have perhaps not enough familiarity with the repercussions of this degenerative disease. And I'm not suggesting for a moment that the regulators slack off, but to treat some of these therapies for really disease that can destroy one's quality of life, that they not be treated as just another small drug application.
So I think, again, the big problem for the regulators, and like I say, the FDA are very good, don't get me wrong, the FDA are very good. The MHRA in the UK is very good. The committee I was alluding to in terms of sticking the cells in the leg has now been abolished, which is very good. I mean, we really went through the mill, and I think part of that may be because we were with such a large pharmaceutical company as well. Pfizer believed that for sure. But I think because now there are, and probably specifically in the eye, a number of cases now, I think that risk is, it's all downside for the regulators, to be honest. They're in a very difficult position. If this works, no one's going to talk about the MHRA being great. Okay, I definitely won't talk about the one that got abolished being great, that's for sure. I think the regulators are good at this when sense is seen. They realise the clinical problems which are associated with this. But you do need that patient advocate component as well. You definitely need the charities, the patient advocates, the hospitals, really telling them that this is a big unmet clinical need. This isn't small. This is huge.
Hi,
I
Kim
would
O'Connor.
just,
I'm
sorry, I just want to add to that a couple of things that, in our experience, again, very early on, we've had good interactions with the FDA, but occasionally issues have been raised, and we've got FDA consultants who've given us different advice, and it would have been helpful to have the opportunity to ask more questions beyond the number of meetings that we're allowed, to have more of a conversation about some of these issues, because so much is riding on it. We can't afford a redo. And so I think that if we could find more avenues for information exchange with the regulators, that would be incredibly beneficial. Yeah.
I must say that does, on my side, seem to be a big difference between European and US. It's much easier in Europe to continually go back and ask for meetings with the MHRA. I mean, you saw that panel where we'd met God knows how many times in the end. I mean, it isn't the norm in the FDA. You do your pre-IND, and then you may, have a couple of chances after that. So maybe, and I know, obviously it's because of the load the FDA are facing. Presumably they have a huge load. So, but it is a big difference between European and US.
It may be it's sort of an interesting challenge. We've been thinking about it at the Michael J. Fox Foundation too, but with Parkinson's, and different ways to engage the FDA and regulators in different conversations. And obviously, if it's a very specific drug program, you may have to go through the more sort of traditional channels. But if there are common sort of questions, sort of bigger issue type questions that you can engage them on as a group, it's often very valuable to have, and they seem to be very open to this. You often have to have it here in DC because it makes it easier for them. Workshops on very specific topics, and we've done this a few times already in Parkinson's, looking at better scales for cognitive impairment in Parkinson's and things like that. And they're actually very open to this, and they're very willing to come to those types of meetings. And so if you can kind of frame the challenge you're facing in that kind of sort of more open framework, that's actually another option to have sort of more, as you say, more of that sort of direct conversation, I think. So it's something we're definitely exploring.
And in California, CERM's been very good at this in terms of engaging with the FDA and having the group be involved as well. So, CERM's very proactive in this area as well.
Kim O'Connor, Tulane University. This question is for Peter. On your procedures, did you see any secondary eye trauma or disease, both transient and long-term, in terms of cornea, edema, pressure buildup, in terms of glaucoma? You mentioned you're putting steroids in.
Yeah.
What did you see?
So the big issue with using a steroid, as you know, anywhere near the eye, is the possibility of getting increased ocular pressure. Mm-hmm. And no, we haven't actually seen any issues with increased ocular pressure. The second patient was diabetic, and there was issues with his diabetic management, but that was mainly due to the fact that he wasn't managing the diabetes very well. So, but that was sorted with him. But no, there's been no adverse effects whatsoever from the actual procedures themselves. Interestingly as well, these were very, very, very bad bleeds, and we've not seen any bleeds post the implantation of the patch, even though there's no anti-angiogenics being given.
And I just wanted to make a couple comments. In our AMD trial, we also didn't see any adverse events of the transplant procedure, although we weren't putting in a patch, we were putting in cells. But we did, since these patients tend to be older, they're less tolerant of the immunosuppressants as compared to the children or the patients in the spinal cord who tolerated immunosuppressions quite well for an extended period of time. So,
just a quick question about time. How long do we have for the questions until 2:50? Okay, great. I had actually one question sort of about cell sources and sort of the rationale behind the different types of cell approaches you've thought about using, and it was interesting to hear both Sally and Anne sort of talk about their approaches, which for you actually isn't really so much a stem cell based, it's more, I guess, primary tissue cell based. And yours is sort of fetal tissue derived approaches, and some of the, I think, pluses and minuses of that. I'm just curious, maybe touch a little bit more on kind of the thinking and the rationale that goes into a decision about going for that type of approach versus sort of more sort of the stem cell based approaches that many are using. And either.
So, we started the work on the fetal-derived neural stem cells before the human embryonic stem cell was found or discovered, or the iPS. So this started way before then, and at that time the thinking was, because I had worked previously on the human hematopoietic stem cells, purifying those, that we could do the same for the brain, the liver, the pancreas. Frankly, the easiest one to identify and purify was the brain stem cell, so we ran with that, because within a year of starting the company we found that cell and could expand it, and then when we found the biological properties that it could make all the 3 cell types, granted we didn't pursue Parkinson's because it didn't make the right dopaminergic neurons, but then it's a matter of choosing your targets. So we focused on myelin disorders and traumatic injuries like spinal cord and the dry AMD. So that's the rationale for ours. It's more like the timing of the science.
So for us, we were actually inspired by prior work that had demonstrated that the retinal pigment epithelium in some organisms like salamanders could act as a stem cell that would regenerate the complete RPE and the neural retina in about 4 weeks. So in these animals, you can remove the neural retina and the retinal pigment epithelium will reactivate, change into a precursor that produces that. It's really quite extraordinary. And we began to think about what regenerative potential might be present in the adult and in the adult human. And so we went at this looking for the possibility of a stem or progenitor cell there. And what we found was that there were cells with stem cell properties there that are self-renewing and multipotent. We've done that by clonal analysis, by time-lapse microscopy. We've identified the subpopulation of cells. And so it's a little bit different from primary RPE because most of those cells actually don't survive the growth process that we do. What we're working on is a tissue-specific adult stem cell product. And because we can isolate it from eyes, we think that an exciting possibility is to use it to see if we can activate it to get more regeneration from the retina as occurs in salamanders. So we're actively working on that. We've also been able to retrieve these cells from patients with AMD who have a healthy peripheral RPE, and we think we might be able to, well, we've got them also from living patients who then, in theory, we could use to build up an autologous transplant. So it really was inspired by basic biology, but it's led us down this very interesting path.
Sally, Steve Rose Foundation Fighting Blindness. So you're calling it a retinal stem cell. How would you say it's different from the retinal progenitor cells that have been characterized and in fact are in clinical trial at the moment? That's the first question. Second, building on the salamander work, have you all done a proteome, all the analysis necessary to understand what that process of rebuilding the retina is, and looking at that with respect to how you're using the whatever stem cell progenitor cells that you're isolating from the RPE in order to drive the process, meaning that you're producing RPE, but could it in fact be producing other retinal cell types as well?
So are you referring to Henrik Claessen's cell, which is... Are you referring when you say,
well,
how
the
does this cell...
neuroprogenitors, whether it be Claessen, whether it be
Okay.
Mike Young's work,
Yeah.
whatever, they're retinal derived?
Yes, they are. So those cells are derived from the fetal neural retina. They are retinal progenitor cells that are poised to make neural retinal cells, the ganglion cells, photoreceptors, horizontal cells, etc., of the neural retina. By that point, there's a divergence away from precursors in the RPE. The cells have made that bipotent choice. And so those cells do not readily make RPE cells. So the cells that are quite different that we work on because they're made within the RPE layer. And we're very interested in the plasticity of these cells. We have looked at many characteristics over time to see if we can get them to produce retinal neurons. We have finally been able to reprogram them, but we do that through the introduction of transcription factors. And so that's something that we're very excited about pursuing that, actually. Thanks. So we haven't done that. I know that was a difficult analysis for a long time because of the size of the salamander genome, which is ironic.
So Steve, we've done some of that work where that effect in salamander is still present very early on in mammalian RPE. So there is, you know, a timeline there, even within the mammalian retina, where you can actually see regeneration from the RPE. One of the big issues, as you rightly say, is so when that stops, why does it stop, and what has changed in that particular phenotype of the cell? So yeah, there's a fair bit of work ongoing now to try and look at that.
Yeah, and Ellie Tanaka is also working very actively in that area. It's an interesting question.
Yes, Seigo Izumo of Takeda Pharmaceutical Company. It's not covered in today's presentation, but stroke is a major cause of chronic care dependency. What do you think the potential of a regenerative therapy for stroke?
So Reneuron are just reporting on their trial where they've used the cell therapy for stroke. They were, again, very severe cases, a number of years post the initial stroke. And they are reporting some interesting findings, you know, in terms of the MRI, etc. Their cell therapy is based on the cells inducing a regenerative region near the stroke. So the cell itself dies. In fact, they don't use any immunosuppression at all. But it seems to cause some sort of endogenous activation. So similar to what Sally was saying around if you can actually activate something in vivo, then that would be a great therapeutic. And like I say, the work was done in not Edinburgh, it was done in Glasgow, I think.
We've done some work with Gary Steinberg's lab at Stanford on stroke, and in the preclinical animal models show that there can be some benefit of transplanting these cells. The cells migrate preferentially to the stroke lesion if you transplant it outside the lesion, and they migrate toward the lesion, and that there are several properties of the cells that might be beneficial in the stroke. We haven't pursued a clinical study because, you know, the complexity of doing a stroke trial is very vast, but hopefully somebody will pick up this technology and pursue those other indications.
Thank you.
Maybe one last question, and then I think we'll switch over to the next panel. I'm just curious, sort of, when we think about sort of the differences between targeting an injury issue versus a degeneration issue in something like neurology or macular degeneration, where there's obviously an underlying disease process that's continuing, presumably, even though you're sticking something new in there, versus obviously something like spinal cord injury when you're just trying to repair what's been damaged. I'm just curious, when you think about your strategies here, sort of how do you deal with this? In Parkinson's, there's some at least evidence to suggest that the pathology can actually sort of transfer into the new transplanted tissue based on some of the fetal tissue trials. And I'm just curious if this is also in your thinking about sort of the long-term, I guess, viability of some of these approaches.
So in our spinal cord trial, we purposely didn't go into the acute phase because we wanted to be in the window of time beyond any spontaneous recovery, because in the acute phase, you're having a lot of immunological inflammatory processes going on, and we didn't want to be in that window. And we wanted to be confident that any restoration of function that we saw was going to be likely due to our cells rather than a spontaneous recovery. So we purposely chose a time window that was beyond the acute phase of the injury, because we know that the inflammatory processes were probably very harsh on our cells. So.
What you're seeing in Parkinson's disease with the fetal transplants which have come to pathology is, you can see Lewy bodies. So in the cohort of patients where we use their own cells, they've gone 8 years now, and those cells are still surviving and they've got good vision. So and the actual condition itself takes about 65 years. So, neurodegeneration, if you're putting in a zero cell, aged cell, then the possibility is it won't be affected for a number of years, if not the remainder of the life of that individual patient.
Yes, and we've found that when we reactivate this subpopulation of RPE stem cells, we reverse several aging markers that are obvious in the primary cells from the RPE. And so we are also hoping that in doing that, we will rejuvenate them, and as Pete says, that the cells will have several decades of productive life in the person. But I would say that your question about the difference between injury and disease is extremely important. There are no animal models that accurately model macular degeneration. Mice don't have macula, so not a good animal model. And therefore, that unknown of how the disease environment will affect the cells is something that I think will all be, that's something that we will have to look out for.
So, I think Sally's point about understanding the disease model, people had said that in Alzheimer's disease that the plaques were toxic, but we had done a study where we transplanted our cells into several Alzheimer's model and show that the cells survive directly adjacent to the plaques and there's no damage. So, and we in another model we saw that you have an increase in these synapses as compared to the non-transplanted control. So, I think it depends on the accuracy of the disease models and the types of cells and, what is really the causative agent of these neurodegenerative diseases.
Okay, thanks so much to the speakers.